Good evening, everyone who are based in Australia, and morning probably, or afternoon for those who are outside. My name is Pyre. I'm the CEO of Smart MCs, and today we're very lucky to have a special guest, Reza Nosrati from Monash University, who's doing a fantastic research in essentially biomedical engineering interface with biology around engineering reproduction. So without further ado, I will pass on to Reza, where he can explain, give a quick introduction of himself, and then we'll have some cool follow-on questions on his academic pathway and some probably good tips for those of you who are a bit earlier throughout your career. After you, Reza. And by the way, this call is recorded. This video conference is recorded as well, and we'll upload it on YouTube after the seminar. Thank you, Reza. It's all yours.
Thank you, Pyre, and hello, everyone. Thanks, first of all, for the invitation to speak as part of your seminar series, which is very exciting. I guess, as you guys can see, my background is in mechanical engineering, but I'm mainly doing application of microfluidics, machine learning, and advanced optical microscopy system for reproductive biology, which is a very interdisciplinary field. And since Pyre has asked me to talk about my research and also some of the lessons I've learned about my academic path, I think I'm going to give you a very quick overview of some of the part of the work we're doing, mainly the works that involves a lot of different techniques, and it's a bit more interdisciplinary. And then at the end, I kind of switch to say and talk about some of the lessons that I personally learned moving through from my PhD to postdoc and to my academic position. Hopefully, I guess some of those lessons would be very useful, especially for more senior PhDs and postdoc, but also, of course, I think more broadly for people outside of the academia. I guess the key motivation for going to small scale is when we think about biological systems and different phenomena happening, in general, most of these systems include interactions, either cell-cell or cell surface interactions or reactions happening at the scale that is beyond what we can resolve using clinical sample length scale. When we think about clinical sample, we're usually sitting in millimeter or higher, but then cell-cell and cell surface interactions really move here, which is in micron scale and even can get to a smaller scale. That's a very good opportunity for microfluidics, because then we can build environments and chambers where we control the environment very carefully. In most cases, we can provide visual access. Then it means that if we link it up with advanced microscopy and some careful analysis and processing, we can extract a lot of useful information. And that's what we try to do in my lab. I wasn't sure about the audience, and I'm also giving a very quick overview of microfluidics. It's essentially the science and technology of controlling and manipulating small amount of fluids, usually micro to picoliters. It can be both in the format of paper-based microfluidics devices like what you see here. These are mainly the application, especially in the biomed, is for low-cost diagnostics. They're very easy to manufacture, scale, disposed of, and especially when it comes to biological samples with concern around contamination, that's quite useful. But for most of the things that I'm going to talk about, essentially we use these kind of traditional microfluidic devices. These are micron size or even smaller size features that are patterned in different types of substrate. In most cases, we look at PDMS or glass because they're transparent and give us good visual access, and that help us to combine it with microscopy for single cell resolution. In my group, we look at opportunities when we can apply these techniques for diagnostics and sperm analysis. This range from paper-based strategies to more recently application of AI and in particular machine learning. A big focus of the group is on fundamental understanding of sperm motion and reproductive tract function. This is where we learn how physiologically relevant changes in the female reproductive tract environment influence the fertilization process. And then eventually we use these two knowledge to kind of develop new high throughput sperm selection technologies for treatment application. Just because of the time constraint, I will not touch on the third subject, but I will very briefly review some of the, I believe, interesting and exciting project that we have done in this other two area recently. I will not go into details, but I'm happy to answer any question later on by email if you want to learn more about these works. When it comes to sperm analysis, I guess our group, like many other groups, has started to think about replicating some of the existing practices, making them more accessible and affordable, and provide opportunities for low-cost diagnostics. An example is this paper-based device, which is essentially a colorimetric assay that when you apply a droplet of semen sample into this concentration and motility spot, you get a reaction between samples with metabolically active human sperm that they have NADH plus. The NADH plus will basically react with the tetraxyllium salt, which is yellow sitting here. And then based on how much of that NADH plus we have in the sample and the concentration of live and motile sperm in the sample, you get a color change from yellow to pink. And then if you can detect it with your eyes, that patient is within the safe range in terms of fertility potential for concentration and motility. And if you cannot detect it, then that person needs to consult with an IVF clinic. It's a very good device. It's low cost. I guess has a very good commercialization opportunity, but still we're not doing anything to really help improve practices and remove some of the barrier when it comes to infertility treatment. And I believe if you learn a little bit about some of the practices in IVF, which is very similar to some of the other clinical practices, they're very manual, they're beaker-sized processes at the population level. Then they're really not looking at single cell and individual cell, which is, I think, a big problem when it comes to assisted reproduction or reproduction in general, because I think that's one of the most fascinating events happen at the single cell level. Then we really need to inform this practice with single cell level information. And that has been a motivation for us to move away from those type of applications and start thinking about how we can use microfluidics in combination with machine learning to achieve two main goals. One is we want to move away from sample level analysis to enable single cell level analysis. And then while doing that, we want to avoid a number of manual and subjective steps in the process. For example, when we think about morphology analysis, currently it's being assessed manually by visual observation under microscope, takes a lot of time, very subjective. And one of the other area that we're interested in, some of the intracellular characteristics like DNA quality, acrosome states, they're currently being assessed by staining, but we want to see if we can replace those staining practices with virtual staining practices using machine learning. And this way we can non-invasively evaluate those parameters, which would be an opportunity then to bring them to the treatment side rather than diagnostic sides, and actually have a better understanding of that one individual sperm being used to fertilize the oocyte. Just as an example, when it comes to morphology, the traditional method is they prepare a slide like this, which calls a smear slide. Then they need to manually look at around 200 sperm and then classify them to normal or any of the normal classes. You can imagine how time-consuming and labor-intensive this work is. And of course, these cells are stained. And it's a really valuable tool when it comes to diagnosis, but you cannot really apply these methods when it comes to the actual treatment side. And we wanted to remove that barrier. We first developed some machine learning models like many other groups, which were looking at these smear slides images and then try to do classification. We were able to develop a model that can predict sperm head morphology from smear slide with 98.3% accuracy, which is, I think, the highest achieved so far. But still, we have the same problem. These are dead cells, they're stained. It's really not useful for treatment. And we wanted to switch to models that can be used. But the problem was there was no training data set, because when you want to develop a learning model, especially the type of supervised learning model being developed so far in the AI for reproduction applications, you rely on your training data set. You essentially need to have, for example, a library of sperm images labeled by expert, and then you use those images, show them to your learning model to say, this is a normal cell, this is abnormal. And then the model start to learn that correlation, and then that's how the model can start to make a prediction. The problem here was there was no data set available. Then we needed to build our own data set to achieve that. We built this model that look at microscopy images, identify sperm in those images, and extract individual sperm images from those. microscopy images, then this way we could build a library of thousands of individual sperm images. And then we built a kind of like a labeling software that our clinical collaborators could use to look at each of these sperm images and then classify them based on head, midpiece, and tail characteristics. And then we use that data to train our morphology classification model. The model we're using use an ensemble structure rather than the typical structure that is mostly being used in these type of applications. The difference between ensemble and the normal model is in ensemble, we're running 4 models in parallel. Here you can see the 4 model, and for a representative image, you can see where each of these models is mainly focusing on. This way, by using the 4 model in parallel, the overall model will have a better holistic understanding of cell biology. How this ensemble structure works, you have an image basically converted to numbers, goes to each of these models. Each model make a prediction about whether this sperm belongs to a normal or abnormal class. They pass on their prediction to a meta classifier, which gets the 4 votes essentially, and then make the final decision about whether this sperm is normal or abnormal. Using this approach, we developed a model that can look at live unstained sperm and more classify it based on its morphological characteristics with 94% accuracy in real time. And this is a unique opportunity. Now we can really take this, and if the embryologist picking an individual sperm for using XC, we can actually in real time say whether this is a morphologically normal or abnormal sperm, and then can help with that selection. One of the other criteria which is important to keep in mind is when we move between clinics and between different imaging settings, you have variation in the capability of those equipment, and you want to make sure your model is not too biased with respect to the original settings that we used to develop the model. That's where another unique capability of the ensemble structure come across. You can see that here we're moving to images that are basically are of lower and lower resolution. And while each of the 4 models will have a significant drop in performance when you move to lowest resolution image, we can see that the ensemble model can kind of compensate for that loss in resolutions and then bring back the accuracy. And we can still predict performance with around 80% accuracy. Then that's great. Then we can do morphology classification. As another example, we also look into virtual staining models. Here, for example, is an example of the model we're developing to predict acrosome state. How this model works is we are building a library of sperm images with the corresponding staining images of that same cell. Then we use a generator. The generator is essentially being trained to just look at the, in our case, the normal DIC image, and then from that predict a virtual staining image. In this case, ours is in FITC channel. And then they pass this image to a discriminator. Then discriminator have both the generated image as well as the ground truth, which is the actual staining image. Then compare these two. And then based on the differences between these 2 images, give back suggestions to the generator to say, this is how we can improve your performance. And eventually we can have a model that look at just the morphology and then predict this virtual staining image. Then we are testing these models. We use a different approaches to test. We do pixel by pixel comparison. We look at histogram, but of course the main criteria is we will look at classification performance of our model when comparing what class the actual image belongs to as opposed to the class predicted for the virtual staining. And based on that, now we have models that can look at acrosome stain and DNA and then predict their quality. And now we're moving to also have central staining as well, which would be very exciting. Then we're trying to bring some of the single cell capabilities from AI to the practice, and we're kind of focusing on the therapeutic side rather than the diagnosis. Now, moving on, I want to also talk a little bit about our work for sperm research. This work is mainly motivated by the natural reproduction process. For sperm to achieve fertilization, they need to basically swim through the female reproductive tract. It's quite a long race, very complicated, but a lot of complex phenomena happening. The ones that we are mostly interested is rheotaxis, and essentially flow and change in viscosity of the fluids as the cell moves through the female fallopian tube, essentially this later part of the tract, to achieve the site of fertilization. That's one thing we're focusing on. And then also we strongly care about how the geometry of the female fallopian tube, and this is example cross-section of the tube that you see here. This is at the port of entry to the fallopian tube uterotubal junction. It's essentially just one opening hole. And as the cells swim through this section, you can see the cross-section becomes much more convoluted, much more confined. And we want to understand what's the role of this geometrical complexity and confinement, which has been largely ignored to date. To be able to understand a lot of these cell surface interactions, what we needed to do is we needed to have a better understanding of the swimming hydrodynamics. One of the challenge was to get hydrodynamics, you need to do micro PIV. To see the cells, you need normal microscopy. Then in order to achieve that, we kind of developed a unique dual imaging platform where you can simultaneously image the cells in DIC mode, and then the cell is in a fluid which is seeded with tracer particles. Then we image the tracer particles in the fluorescent channel, and then we do correlations to extract the velocity field based on the movement of these particles, which is the micro PIV we're doing. And then we can overlay these two to know where the cell is and how the hydrodynamic flow field around the cell is also changing. Of course, this was important for us because we wanted then to move to near surface interactions. But while doing that, we realized that if you look at the cell here, the sperm head is like a flat elliptical disc. And as the cells swim, this disc turns because the cells swim with a 3D helical waveform. And when we started to compare these velocity fields with the orientation of the cells, we realized that these velocity fields are fixed with respect to the orientation plane of the cell. Then we started to arrange that around the cell. Then you have a sperm, you get these different planes, you get the vorticity field, and start to arrange them around the cell. And we did a little bit of a reconstruction and smoothing and modeling. And then we realized this essentially represents a pair of superhelix flow structure that are attached to the cell, and they turn with the cell as it moves through the fluids. This is kind of the way that the cell then penetrate through the fluids and move forwards. But of course, we can learn much more because now, based on the size and how wide this flow field expand into the fluids, we can learn much more about when is one cell essentially start to feel the presence of another cell or another surface nearby. And this tells us a lot about cell surface and cell-cell interaction. This becomes more interesting when we start to look at cell movement really close to surfaces. For example, here, we're looking at sperm. These are normal bright field microscopy cells around 50 micron away from the surface. Then we switch to fluorescent channels. Again, because the head is flat, as it turns, you have different area of the head facing the objective. And that's why you see this blinking effect here. But then when we switch to turf microscopy, you see these cells kind of in a very different motility mode. Now the head is aligned with the surface, and the flagellum, you can see most of it within the field of view. But one thing to point out is when we do turf microscopy, we are only seeing things at the surface within 1 micron distance of the surface. Then this swimming mode that we see here is very surface confined, and it's a 2D beating because the whole 70, 80 micron length of the cell is really confined within that 1 micron depth of field for our microscopy. This indicates this is a strictly confined 2D swimming mode, which we call 2D slithering mode. We later on showed that this is a very specific types of transition. It happens based on different viscosities, but also when it comes, for example, to human sperm, by switching from 3D bulk to 2D slither swimming, these cells can swim much straighter and around 50% faster, especially at fluids of higher viscosities. Then this is a natural mechanism in vivo. When they increase confinement, they bring in more viscous fluids to facilitate slither swimming, which can enable the cell to get to the site of fertilization quicker. And actually, these cells that exhibit 2D slither swimming, we also showed that they have significantly better DNA quality. Then these cells gain a competitive advantage and they're better cells. And that's exciting. It's a very interesting learning. But when we think about the reproductive tract, it's also not a flat surface. It's a curved, soft epithelial tissue, right? This is an example cross-section of this tissue. Then the next step was we wanted to understand the role of these curved surfaces. And in order to achieve that, we use droplet microfluidics. We encapsulated individual sperm in droplets in a way that the radius of curvature of these droplets match the range of curvature in vivo. And then we started to study their interaction with these curvatures. We realized that when the sperm swim in a really big curvatures, they align mainly parallel with the surface, they swim relatively quicker, and now the boundaries is mainly a guidance mechanism. But as we move to smaller curvature, the cell aligns more sharply with the intercell and with the interface. And because of that, they start pushing against the interface and slow down, and they reduce the velocity a little bit. Now, this direct physical contact between sperm and the interface at a reduced velocity, which means the cell actually spend more time touching the surface, it's significantly important very close to the site of fertilization, because it has been shown that this direct physical contact between sperm and the epithelial tissue is crucial for a process called capacitation, which prepares sperm for fertilizing the egg. Essentially boost up its energy, it capacitate the cells, initiate some chemical changes in the cell, while also initiate hyperactivated motility, which provides the very kind of like vigorous movement for the cell to penetrate the zona pellucida of the egg. Now, then these are kind of like start to link to each other, and this becomes more interestingly when we realize the range of curvature where we see these type of change to attacking motility mode match the range of curvature very close to the site of fertilization. Then it starts to tell us how this change in geometry again triggered the timing of these processes. A lot of these, both the 2D slithering, which had significantly higher DNA quality, and the orientation of the cell with respect to these curvature, a lot of these essentially originate from the flagellar movement. But one of the other shortcomings in the field is most of the sperm motility analysis so far are mainly based on tracking head. And we wanted to also move and consider what's the role of tail, what we can learn about the tail and its moving dynamics. That's why we started to use dark field microscopy. The beauty of dark field is you get a white signal in a dark background, and it's perfect for automated image processing. And also one of the other advantages, it gives you much better resolution to resolve really small features, because sperm tail is around 300-400 nanometers, and it's moving at around 5 to 10 hertz. Then it's really high speed, high resolution imaging that you need to do, and dark field allow for that. Then we can image the cell. We have a code that automatically processes this and then extract the flagellar center line. And then from that, we can do some really nice studies. For example, here, you see cells swimming in fluids of different viscosity. And I think there are two very interesting points here. First is you can see as we increase the viscosity, we get a transition from the 3D helical swimming, where the head is turning, to 2D slithering. Now the head is aligned and the tail beats with a 2D flagellar waveform, which is much more energy efficient. The second point, which is even more interesting, is if you compare these three cells, they're swimming in 3 different fluid viscosity, and this 250 is 10 times more viscous than 20. Then it's a really thick, it's a really tough fluids to get through, but this cell still can maintain the same swimming velocity. Then the 2D slithering is really helping these cells to get through those thicker, tougher environment. The next question is now, is this cell swimming better because it's actually becoming more active and essentially expending more energy, or is it just because the cell is more efficient in delivering energy into the fluids? Because we had the flagellar center line, now we can extract not just the velocity, but also how much power and energy this cell deliver to the fluids. We can see as we move to higher viscosity, the cells start to deliver energy more efficiently into the fluid while maintaining the same level of metabolic activity. Then the 2D slithering is not just an increase in the level of the activity of the cell, but the cell just becomes more energy efficient. Then it shows again how that increase in viscosity provides a competitive advantage for those cells that can switch to 2D slithering. The effect of viscosity, which is largely ignored in any kind of like practice, I think so far, is not just limited to sperm. Because when we started to look at the female reproductive tract side, and when we cultured epithelial cells from the female reproductive tract in fluids of different viscosity, we realized that as we go to higher viscosity, you get more ciliation, which is what you need near the site of fertilization, because these cilia start to form patches and then they start to beat collectively in a synchronized wave to make something called metachronal wave. And this metachronal wave has been suggested to be important in terms of moving the fluids, but also transporting the egg and embryo through the reproductive tract. When we start to look at the movement of these cilia, we also realize, for example, this is beating in 1 millipascal, and then this is beating to 200 millipascal. We realize the beating behavior of these cilia also changes, and they start to beat in a more cohesive way where they can more effectively transport things. It becomes much more obvious if we look at the movement of waves generated by these beats. This is an example of a wave generated when the cilia moves in a 1 millipascal fluids as opposed to 200 millipascal fluids. And I think it's very obvious that you get a very nice waveform that can move and transport things. Then the female reproductive tract, the change in geometry and viscosity, both quite important. This was just a highlight, an overview of some of the research we're doing in the group. We have been very successful in terms of establishing strong collaboration and maintaining those collaborations to bring a lot of insight from different perspectives, clinical embryology, reproductive biology into our work. And essentially, I want to talk now a little bit about how you can establish these type of collaborations. What are the type of things that we need to think about as we move through the career? And just keep in mind, most of these lessons that I'm going to share, some of them come from my own mistakes, but also some of them come from the advice that I've received from my own mentors. Professor David Sinton at the University of Toronto, who is my PhD supervisor, and I still learn a lot of things from him, as well as, for example, Professor Moira O'Brien, Dean of Science at UNI Melbourne, who's also one of my close collaborators. So now I want to switch, and I want to talk about some of the lessons I learned through my career so far. First of all, I think it's very exciting to work on interdisciplinary projects. You know, you for example, as an engineer, it's really cool when you develop a tool that can improve knowledge in another area. But I like to highlight that, especially at the beginning, it needs some time. You need to really slow down. And I know it can be very stressful because you feel everyone is moving quickly. Let's say if you're early in your PhD, but you really need to slow down and you need to learn across multiple fields, and you want to start to collaborate with diverse teams. Now that learning process is important because when you work with interdisciplinary team, you need to start to learn their language and speak their language. For example, as an engineer, when I talk with clinicians or biologists, I need to be prepared to talk their language, and I need to be patient for them to learn my language and speak my language too, because we have all different priorities, different mindset, and I think being patient and spending time and learning things is very helpful, because in long term, I think that's incredibly rewarding. That's really something that can help you stand out, and that's where I think it becomes a lot of fun. This is relevant to expertise. I think those of you especially who wants to move to academia, you need to think about developing an expertise. This means that we need to be known for something, at least one thing, but the best thing is to be known for a few things. And that's where, you know, that slowing down and learning things really deep can be helpful. This means that we need to have a plan. We need to think, especially when we start to think about our academic career, I think it's important to think about what I want to achieve in long term and then break it down into smaller little pieces that can inform the type of paper you need to learn, the type of collaboration you need to establish. And this is not just to stay in academia. I think making a decision about what we want to do next is very important, because, for example, if you want to go to industry, then I would say as soon as you finish your PhD, leave academia. Don't waste your time in academia if you do not want to become an academic, unless it's a helpful thing. For example, some people want to go and do really high level R&D job, which needs them to do a postdoc. Then that makes sense. But I mean, have a reason for why you do everything. And then through that, establish a research vision. Research vision, I think, is not what I'm doing today, but it's what I'm doing today and tomorrow and then the next day all come together to help me achieve something in 5 to 10 years, because that's something that can help me to build more impact and actually go deeper and deeper, and that helps me to also become known for that expertise in the field. One other thing, again, I think it's very important that we start to build our own ideas, and I think as PhD and postdocs, we usually think that we own everything that happens in our projects, and in some aspect that's true. I think that sense of ownership of our project is true, but that's not enough to take you out of that group and then basically give you the opportunity to have your own group. I think you need to be known for your own ideas. Then think about, what can I do to complement that existing work? What's the next step in the field? Then you need to really go one step beyond your supervisor. And I think that's a very important thing when it comes to establishing your expertise too. Okay, I don't know if my slides are getting to, yeah. Okay. One of the other things I want to talk about is accept. I think acceptance is very important when you take any role, especially I think in academia, we need to accept our role, our position, and all of the responsibilities and the workloads that come along with it. I'm saying that because you know, you can become that individual. Let's say I can become that individual that I'm always not happy about how everything is being thrown at me. Or I can become someone who accepts my role, think about how I can manage things better. And then I think that's very helpful thing to progress better, but also have less stress. Because if you don't accept your job and its responsibilities, it very quickly, especially I think in academia, it becomes very overwhelming and you will not be happy. And when you're not happy, you're not performing well. Now, it's I like to highlight that this doesn't mean we want to go back to and work crazy. I'm strongly against that. I think work-life balance is quite important. I'm a very 9 to 6 type of person, except for this talk right now, which is fine. But I think it's more about setting realistic expectations and setting a limit for our ambitions. What I mean by that is, if you are someone who really wants to move quickly and really wants to, you know, jump steps, then you need to be prepared to have less time spending for other things. Maybe you have less time even cooking, maybe you need someone at home to help you cleaning. Then these are the prices you're going to pay. Maybe you need to spend more time in the office. But accepting that this is my own decision and I'm doing that, I think it's extremely important. Now, do that to enjoy your work, and I guess I'll stop there. And then think about all of the other things. When especially you stay in academia, I think the success is not just about publishing papers, especially when it comes to your own institution and your position within your institution. I think you need to understand the culture of the institution, all of your colleagues, and all of the admin work and teaching around your work, because those are also important, especially when it comes to promotion and probation. Then try to accept them, try to understand the requirement, and then be strategic in terms of what you do. Whatever you do needs to help you. I think that should be the rule to make the decision. When you make a promise to deliver something, especially at the beginning, do your best, really deliver it well, because that's where you make the first impression. And I think that's very important. And I think a lot of academia, and I made that mistake myself, that when we start, we switch to doing a lot of long hours, but we need to remember that long hours also means you know, a drop in productivity and efficiency because we're just simply tired. Then think about that and then keep that in mind. I think one other thing that is very important is always have the right that it's not only that you need to accept everything. I think your institutions and your colleagues and your collaborators need to also accept you. And a simple example is, if you have family responsibilities, or if I have family responsibilities, I don't need to apologize. That's simply my life. And the only thing is, I need to just communicate that, make it clear, and then go ahead with that. Then then have both sides be direct and straightforward about what's happening, and make sure you deliver on your commitments. I think identifying opportunities is also very important. I think I say that because a lot of times we think people are gonna go and really dig our CV to find how well we are doing in our field, or people gonna reach out to us to say, you know, do you want to apply for this award or put this abstract into this event? That's usually not the best strategy. I think the best strategy is we need to think about what we're gonna achieve this year to help us achieve that bigger strategic goal, and then move towards that and then work for that. This is especially important when we want to apply and target some of the bigger grants or some of the awards, because sometimes they have a special criteria. And if you just want to look at them a week before submission deadline, you may realize that they need something that you don't have. For example, they need some kind of a work beyond your research. Sometimes you need to show some professional activity, some professional development, some community engagement. Then if you plan ahead, you can accept those responsibilities that will help you to check those boxes. And then when it comes to the type of the award, for example, you can meet all of the criteria and then be successful there. Then that's where we need to think about opportunities and match that with our strategic vision to achieve them. And then make sure to plan ahead, have a draft ready well ahead of the deadline. I found that extremely helpful. Get your draft. Let's say if you have a grant proposal or an award application, get it to be read by, like, basically share it with as many colleagues as you can, and you believe that they can provide you a good feedback, and then make sure that you kind of apply those changes. One of the other thing is, a lot of time we travel around to go to conferences, but I think if there's an opportunity to present, take it and then nail it, because that's very important, especially at the beginning. Again, the first impression goes a long way. I also like to highlight the value and importance of mentorship. I think if you want to talk details about mentorship, it's going to be a very long conversation, but you need to mentor others and practice your supervision as you move through your career, but also you need to have great mentors. When it comes to mentors, I'd like to highlight just briefly a few things. You need to have good mentors, and good mentors are people that you like to become someone similar to them, because then you would trust them and you would listen to them. If you have a mentor that you don't listen to, just change it. I think mentor is someone that I know their advice is helpful, and I will listen to them and I will think about their advice. The other thing is, when you interact with your mentor, keep in mind that they're also having their own busy life, and then make sure that you give them enough time to provide their feedback. And most importantly, you give them enough time that when they want to provide you with their feedback, they feel, okay, they can actually do something about my feedback. An example is, if I share my grant proposal with my mentor, let's say 2 days before the deadline, then they know I'm not going to do anything. And probably they're going to just go very light, and their comment may be not as helpful. If I give my proposal to them a month before, then they give it back to me 2 weeks before the deadline, and they know I have a good amount of time to improve. And I think that's very important. A lot of time we miss that, and then we don't get the feedback and improvement that we need. Network and collaborations. I think especially a lot of us within Australia, my observation has been that a lot of times we just think maybe too much about what's happening within Australia, but a lot of time I think it's good to think globally and think about expertise and opportunities outside of Australia as well. When we go to conferences, I think that's a great networking opportunity. Don't be the person who go to conferences and then do their talk or their poster, and then they just hang around and they don't know why they go to that conference. I always like to go to the conferences where I know one of the keynote speakers is someone who I really like to meet or connect with or talk with, at least one person. In a lot of cases, you can find a lot of events which have great list of the speakers. Then make sure to go to those events. Make sure to have a plan, which talk you want to go to, who you want to speak with. If there's an institution nearby, or if within that city there's a really good research lab, send an email, go and meet them. You would be surprised how much of a good impression you would gonna get. And then one other thing is, especially when it comes to collaboration within interdisciplinary team or within the research group itself, sometimes it's easy, and it's like sometimes we get stressed and get nervous about our progress, and then we worry about how much we collaborate with the other team member. But I usually find that to be not a very healthy mindset, and it can impact progress. And I would say the priority is to work as a team, communicate all of the challenges, and then celebrate success. We want to celebrate our win. We want to celebrate all of the competition that we get through, because that gives us motivation and energy. Anything I'm missing here? Okay, and then I think one other thing is time management. I think if we have that long-term strategy, that's very helpful thing to help with our time management, because you want to use that to say no to things. Then you have a very good reason to why I say no to this thing. For example, if you want to give a talk or if you want to go to an event, you go to those events or those talks because you're exploring other interactions, collaborations, and opportunities out of that. If there's no mutual benefit in a collaboration or interaction, that's usually why they die. Then this strategic vision and then matching that with your collaborators, it's a very good way to do a better time management and survive the interaction and collaboration long term. This can also help with some of the, when you move or when you change institution or when you take on your new academic role, it takes a lot of time to establish your lab, set up things. Then that collaborations, which is happening in your collaborator's lab, it's a good way to make sure your publications and your track record is basically not impacted so much. And one other thing is, I think time management is the key to a stress management. We usually get very stressed when we have a lot of things to do. And if we can manage them, we know we can handle them. One of the big lessons I learned as an academic, especially, is try to practice not reacting quickly, especially if the situation is very tricky. I would say the best approach is avoid making decision right away. Step away, try to cool down, think more about this, talk with a friend, talk with a senior colleague, get advice from your head, from your dean, and then react and decide what you want to do. This is also go back to your mentors. I like to highlight one thing about mentor, which I forgot. When you have mentors, try to have two types of mentor. You want to have a mentor from your own institution because they understand what it means to be a good citizen of that institution, and then they can advise you in that line. But also you want a mentor who's outside of your institution, because then you can talk more freely with them and then get better advice from them for some of the more sensitive topics. And then this don't react thing is sometimes you're sometimes I think when I especially talk with some of the other colleagues, you feel like they're in a situation that they don't really enjoy anything. They're not happy with their work. They're not happy with their environment, but they just don't do anything about it. I think that's the worst thing to do. If you are in an environment that you feel there's something wrong, either think about how I can fix this and move forward, if that's a very good environment, but you're just dealing with that one little thing. Or if you feel there's so many things wrong, then maybe your plan should be, what should I do next? Where can I go? How can I move to a different institution that change these things? Then, I mean, again, don't waste your time just doing nothing, make a decision and then act based on that. And then one last thing about this thing is make sure to switch off. I found it extremely helpful to have a day that I just don't check my emails, don't read anything, don't respond to anything, and just step away from my work at least one day a week. For me, it's, let's say, Sundays, but I mean, make sure you have that because you really need to recharge. I will finish by highlighting a few things about group culture. I think it's essential to have a vision and system for the group. For example, my approach is I usually set really high standards, very demanding, but I also try to be very supportive and then provide the resources that they need. That's essentially based on the supervision I also received, and I found it to be very helpful. I found it to be very important. Sometimes we set expectations for our students, but it's not very clear. Then I find they get very confused. Then I always find it helpful to set expectations. If you can develop, for example, a lab manual for your group, do that. That's a very helpful to establish all of the criteria around these different topics. collaboration is important. And then every opportunity is a learning opportunity. Then when you comment on papers or when your supervisor comment on the papers, learn about that. I think mutual respect and interaction among groups is also important. That helps with having communication, for example, in the group meetings, and not judging each other, because that's how people will feel safe to share their knowledge and then get feedback. And again, I think I mentioned this, I think celebrating wins and having a happy environment helps with a lot of things. And also make sure to encourage work-life balance. A lot of time we're especially as academics, we leave, but we don't know how much pressure our students put themselves through. Then I think it's a very helpful thing to make sure, they leave when they need to, they take enough break, and then they also recharge. And if you set any rule in your lab, make sure you follow it yourself, because you need to become the best example of how you want your lab members to act. I hope you find some of these comments helpful. Sorry if I talk too long, and I want to highlight that I think everyone has their own story, and there's no one solution fit all here. It's I found the most difficult part of our job to be interacting with other individuals, because that's the nature of biological diversity, which means that a lot of things are different, and then we just need to understand that and then try to find a way in between. Thank you very much again for this time, and I hope you find it useful.
Thank you so much, Reza. That was a fantastic talk. We have a couple questions. I think you must be a bit of brain reader, because I was just having a few questions in my mind. You answered them as we go. I have a lot more. But anyways, before I get to those, we have a question. Zoe essentially is asking, she's saying, thanks for the great talk. She's asking on the importance that AI will have on the future of sperm selection. So what do you think that the role of AI will be in sperm selection, particularly, rather than just
a great question. I think what will happen down the road, maybe not too far down the road, but in a few years, is AI can help us to have more quantitative assessment of how different criteria of sperm or oocyte links to outcome. And I believe that will help us to define new criteria for assessing quality. Then, because I've been asked, especially by embryologists, quite a lot of time, that will AI replace us? And I think the answer is no. The AI will not replace them, but I think it will help to establish new criteria, and it helps to make tools that can automate some of the processes, which means that IVF clinics will have the opportunity to invest their time more efficiently. For example, if you have a very good embryologist, you want that embryologist to deal with the most difficult case. And I think AI can help there. I think those are the two main near future developments.
I think it's fantastic. And given all the progress that's happening in AI, I think that's certainly going to be helping clinicians for sure. One question, I mean, you spoke a lot about the male reproductive system and sperms, how the emotions affect, etc. My first question was, you didn't do much on the females. You had a few slides then at the end, which kind of got me very interested as well. How are they different? Is the female reproduction system as explored as sperm side of the story and the male ones, or is it less explored in the world? How's it balanced?
I think they're both quite, when it comes to reproductive biology and clinical research, I believe they're very extensively studied, and there's a lot of interesting work happening. When it comes to kind of biomedical engineering type of applications, especially when you go to embryo side, I believe there's still room to do much more work. And essentially a lot of that is because you need a very strong ethics to be able to do those type of work. Then I think that's a very natural barrier. But a lot of work are being done there. Especially when think about AI, I think a lot of AI work we are currently on the egg and embryo side are based on the EmbryoScope, which automatically take time-lapse images of the oocyte and embryo. I don't like that approach quite a lot, because I think, especially with AI, you need a very careful controlled data collection. And maybe it's a good time to start thinking about, you know what, I really need to move into the direction that have very targeted type of studies. Then I think there are a lot of studies out there and a lot of interesting stuff being done, but maybe we need to change our tactic a little bit. And one other thing is when it comes to rheology, that has been largely ignored in reproduction, even more on the egg and embryo side. For example, we still do embryo culture in water-based media. We still do not monitor the anatomy of the female fallopian tube or the reproductive tract. While these things can quite significantly contribute to infertility, then maybe there could be reasons for some of the unknown infertility cases.
And do you see it like with potential innovations in the field? Are they going to be more on kind of the fundamental side on studies with fallopian tubes or the rheology, for instance, that you mentioned? Or is it going to lead, do you think, also besides just fundamentals to some like technological outcomes, like using that to get a better reproduction like outcome or something?
That's a great question. I think any science needs to make its impact to be useful. Then I don't think anything wants to stay with the fundamental side, and the goal should always be I learn the fundamental, and then based on that I develop a technology, and then I change something. For example, in this aspect, when we started to see how viscosity influenced both the sperm and the female reproductive tract, and now we're working on a kind of like a follow-up project where we look at how it influenced embryo development, how it influenced endometriosis, how it influenced the female reproductive side of fertility. And we started to do some clinical work to evaluate their importance. And then if the results are positive, we think that we can now move to the next generation of culture media, which are more viscous, and then when it will have its clinical impact.
That's very cool. Yeah, no, that was good. And one question, I felt like from based on your talk, everything was around human sperm and reproduction. How much work has been done across different species in literature and within your group? And also, is it like a completely different story, like when it comes down to like mammalians or even non-mammalian species, or it similar type of like movements, like sperm motions and then eggs and how the whole fertilization works?
The work are quite extensive, and they kind of expand across multiple species. In my lab, we do a lot of mouse, bull, and human. Human, of course, the clinical impact is quite obvious, and that's why we need to do those works. Sperm is slightly different when you move from one species to another species. Sometimes it's even very significantly different in terms of their morphology. With mouse is a very good model for looking at human embryos, for example. That's why we do a lot of those works. And also we can establish knockout models to look at gene abnormalities, and then this is why we do that. Bull has a good morphology, which is not too far from human, then it's a very good kind of proof of concept before doing human work. But also there's a lot of other works, especially when you think about evolution, endangered species and things like that. There's a lot of research on their sperm and then their behavior, how pollution, change in climate influence and impact their behavior as well. And it's a massive body of work that expand a lot of different applications, not just treatment and infertility, but also agriculture and animal breeding. And that's why it's very, very broad.
Yeah, super diverse looks like.
Yeah.
But that's cool. And then Abhinav asked, so essentially he thanked you for the great talk. I'm glad everybody's sharing that as well. So he's asking if do sperm cells experience shear stress inside the female reproductive organs? How much can they withstand while remaining active?
The answer is yes, they do experience shear. There's always a flow through the female fallopian tube, usually from the fertilization side towards the basically outside of the tract. And sperm has this rheotaxis behavior that they reorient and then they swim against the flow. They can usually, I think shear rate of 3 is the favorable shear rate for rheotaxis, and we have some studies in that area as well, which I didn't share. But when you go to like 5, 6 or higher, then they kind of become not sufficiently motile to overcome that flow, and then they start being washed off by the flow. if that's
But
I
yeah.
think that
That's
answers the
excellent.
question.
Lynn also is asking one follow-up question on the animal side. So he's asking if the sperm selections in the animal world is kind of progressing slower than the human side, or how's that kind of balance happening then?
I think that's a fair comment. I think that's moving slower. But the main reason is because when you go from human to animal, you kind of are scaling up, and that's why the type of throughput that they like for when it comes to animal applications goes much higher, which becomes a little bit of challenge when especially you want to do microfluidic type of selections. The type of environment that those kind of insemination practices being done it's quite like a low tech environment, then you want a really robust, simple technology, and I think those all contributing to have like a much slower progress in that side, at least up to now.
Yeah, that totally makes sense. And in the interest of time, I'm gonna quickly kind of move to more towards the questions on the carrier side of the story, and I'm cautious of your time as well. So Lynn is asking you as an PI essentially, what is your expectation and how do you manage the performance of individuals within a group? I mean, you talked a lot about obviously
Wow,
culture and
very
stuff.
tough question. I think my expectation simply is, let's say if I'm dealing with my student in my group, I think they need to know why they're doing what they're doing, and they need to know the depths of the methods they're using. Then especially if it's someone starting a new field, I think I really care about them slowing down and learning. For example, even if you run, let's say, DNA assessment assays in our lab, and a lot of people run it, but I think they need to know why that assay works and what's the underlying biology of and the function of those dyes and everything. I think that's the way that I try to build that depth of knowledge by being more picky about, how your method works, what does this term means, and over time they go back and read more and more and more, and I think that impact and depth come across, and that helps them to progress better. In terms of monitoring individual people progress, that's really individual. It varies from one person to another person. For example, some people, when you put them under pressure and then when you set deadlines, they perform better. Some people, they get really stressed and then they stop performing. Then I think one of the very first step when you have a new member joining your group is you need to give them some time, and then carefully watch where they're really capable and comfortable and where they are not. I think where they are comfortable, that's where you can push them more. But if they're not comfortable in some area, I think you need to go there much more slower. And it's a difficult thing. I like, if anyone find a solution for that, I would love that.
Yeah, I know. And I mean, human nature is super complicated.
Yeah.
You mentioned each individual is different. And what I found was some people might stress completely about things that others might not, right? So that's where things are very different. I mean, you spoke about mentors as well. One thing I personally found, like, people undervalue a lot the role of mentors, even just like undergraduates, right?
Yeah.
When do you think is a good time to engage with having mentors, and how do you think is the best way to find them? I know it's not an easy answer, but like, where did you find your mentors, for instance, as you went through this pathway?
I think everyone can become a small version of a mentor. Everyone we interact with. For example, I think it's about what I need to learn. For just as an example, sometimes we're just CC'd in a very general email that it's not relevant to us, but it's a tricky email, and then you can see how someone else, which is more senior, reply to that email, react to that email, and we may not ever see any of those individuals in that email. I think that's a mentorship opportunity. That's basically when I'm learning something from that person. Then I think every interaction is a mentorship opportunity. And we need to think about what I can learn from that. But in terms of how I can find a good mentor, I think, of course, academic supervisors, they are our mentor. If you're not comfortable with your academic supervisor, then you can't push it. But I mean, they're always great examples. I would say a mentor should be someone who you feel comfortable spending time having a coffee with them. If your mentor is someone who you really get stressed or really nervous talking with them, and you're worried about how they're going to judge you because you share your concern, I don't think they're a great mentor. They're going to be great mentors with you. I think the way that I find my mentors is my supervisors, people I see in conferences and events, and then I start to collaborate with them. And then, you try them with one thing, and then you see how well they respond and how well they kind of care about their feedback to you. Some people are just too busy, no hard feelings. And if it's not good and if it's not the way that you want it, you should move on. And one other point about mentor is sometimes you work with someone who's actually a brilliant mentor, but their style and their approach is just not fit well with your style and your approach because you may have a different system. Then I think that's also something to think about. I think it goes back again to that human nature. The only one rule I would say is you really need to have them, like when you want to pick a mentor, pick someone who is someone that you want to become. You want to follow their steps. I think that's very important.
And my last question, let me twist this a little bit. Now, from your end as a mentor yourself, what characteristics do you look for those mentees? So, for instance, if a student comes to you, whether it's an undergraduate, PhD, whatever stage, obviously you're very busy. All the senior people are very busy. So, what makes you want to spend some extra time with that person? Obviously, it's always without it. Like most cases, it's not like they're paying you for anything or there's no direct compensation for your time. What is the characteristics that kind of makes you pick those people and work with them?
I think there's two things. I think if it's a good mentor relationship, they take your advice seriously. It doesn't mean they do what you ask them. Sometimes they have their own, we need to understand it's at the end of the day, that individual decision about whether or not they take that comment, whether or not they apply that change. And we need to respect that. But you'd like to see that they have done the work that you have asked for it, because that's how the next feedback going to get better. And then the next time they're going to get advice from me, I spend more time. And I think over time, if they listen and if they improve, then they're going to progress. And then it reaches to a point that you start also learning things from them. Then I think it becomes a two-way relationship. Or they become your collaborator, or they become your grant application body. Then I think that's what I'm looking for, that they listen to me, and then over time, they start to actually teach me things too.
Cool. And is there any kind of things that if somebody wants to come to you and ask you for help, anything that you would kind of be your immediate internal checklist that you would want to say, if I want to help this person or not?
I think, I don't know if I have that. I guess the only thing is, if let's say if someone approached me and then the way that they drafted their email or the way that they drafted that document is something too far from how I do things, then probably I'm not the person well suited for them. Again, it doesn't mean that draft is bad. It's just that system doesn't match, I think.
Yeah. That's, I think, a great one. First impression, kind of first impression, right? So that's cool. Reza, I'm cautious of your time, and I know it's 8:06 p.m. in Sydney / Australia in general, the Eastern time. So thank you so much. It was really valuable kind of contribution you had, and we really learned a lot. And I hope the audience on YouTube as well in future will learn a lot more as well. Obviously, everyone, you can directly reach out to Reza if you had any specific questions, particularly about his research, or if you wanted him as a mentor. That can be, he's a great person. But beside that, that's all from my end. Is there anything else, Reza, you wanted to say before we hop off?
No, I think, thank you very much. It was it was a different experience. It was fun, and hopefully it was useful for everyone. But thank you so much.
Thank you so
I
much
look forward
for your
to seeing you in
time.
person soon.
Yes, likewise. Thank you so much, and thanks everyone for listening. Have a good.
Take care, everyone. Bye.
Yes.
Bye.
Bye.